Welcome back. Let's continue. We were still on the reactions that are taking place at the cathode of both electrolysis of sodium chloride and electrolysis of copper chloride. We said that sodium will not undergo reduction at the cathode because it has got less electrode potential compared to the water. That's why hydrogen gas will be formed. And then on the other one, we said copper will be reduced at the cathode and no hydrogen will be formed because copper ions have got higher oxidizing ability than water. Now they can ask you this question to say why this is happening. So you need to explain for 3 marks. In the case of sodium chloride, we'll say water is a stronger oxidizing agent. Remember, all oxidizing agents are on the left hand side of the double arrow than sodium ions. You get 2 marks for saying this, and therefore it will be reduced if you want to continue to form hydrogen gas. Whereas in the case of copper, we'll say copper ions are stronger oxidizing agent than water. You get 2 marks for saying this, and then therefore the copper ions, it will be reduced to form copper. It will be reduced to form copper. It will be reduced to copper. Okay. And then from here now, once you have explained the oxidizing and the reducing abilities of the 2 electrodes, another question can come and say, now what happens now in the sodium chloride cell, and also what happens in the copper chloride cell? What are the observations? Then sometimes they can ask you to say what will happen to the pH of that solution. Therefore, because the pH of that solution will be greater than 7, since OH minus is formed. Or if they ask you about the color of the litmus, we know the litmus will turn red in that solution because of a base is formed. In this case of copper chloride, we'll know that the color, the observation is that now the color of the solution, will change from blue to colorless. We know the blue ions in the solutions are the copper ions. Copper ions are blue in color. Therefore, that will be the observation in the cell. Otherwise, they can ask you as well to say, why do we prefer DC over AC in the cells? When the cells are operating, why do we prefer DC? We need DC because we want to maintain the polarity of the electrode. To maintain the polarity of the electrode. To maintain the polarity of the electrode. Polarity of electrodes. So we know that if we are using AC, AC is a current that changes its direction constantly, meaning that now the electrodes will change their polarity every semicircle, every 180 degrees. So it will change from positive, negative, just like that. So that's why we prefer DC, so that the polarity of the electrode will remain as is. If it's positive, it will remain positive throughout. If it's negative, it will remain negative throughout. Hence, DC is preferred than AC as a power source to convert that electrical energy to chemical energy, so that you can convert that energy into the chemical reaction in the cells. So that's all about the electrolysis of sodium chloride and the electrolysis of copper chloride, where we are using the inert electrode, which are carbon electrodes. So remember those inert electrodes, they are carbon electrodes. At the anode, there will be chlorine is formed in both cases, whereas the cathode, in the case of sodium chloride, hydrogen will be formed. In the case of copper chloride, copper will emerge. There will be the brown layer on the cathode electrodes at the cathode. So let's pause. We'll talk about the two other cells which are using active electrodes. Thank you.